second objective is to talk about the electrical properties of the heart and pretty much the conduction system, which comprises two types of cells in the heart. Okay, now the heart has this autorhythmicity, and this is the ability of the heart to generate its own rhythm in the absence of any neural input, in the absence of any external stimulus. Okay, so this autorhythmicity, it allows or provides a pathway which allows the excitation of the heart to spread. Okay, this is going to be important. This is going to be what provides the contractility of the heart as that conduction, that electrical activity moves throughout the atria and the ventricle. Now, there are two types of cells in the heart in general. There are autorhythmic cells, and of autorhythmic cells, there are two types, pacemaker cells and conduction fibers. And then there are contractile cells. So I want you to bear that in mind as we talk today. There are two main types of cells in the heart. Your autorhythmic cells, which can generate an action potential on their own. Okay, and examples of those are your pacemaker cells and your conduction fibers. And then there are your contractile cells. Contractile cells are more in number, right? They're the majority, and they're responsible for the actual contractile force, so pumping, contracting of the ventricles. So we'll start off by talking about the autorhythmic cells, which are the pacemaker cells and the conduction fibers. So pacemaker cells have the ability to spontaneously depolarize, and when I say spontaneously depolarize, I mean that they can change the membrane potential. Okay, so there should be something we know already. If you change the membrane's permeability to an ion, you can change the membrane potential and you can spontaneously generate an action potential or spontaneously depolarize. Okay, and as such, these pacemaker cells coordinate the rhythm of the heart, which is the heartbeat. Okay, so the heart is essentially keeping its own rhythm via the pacemaker cells. The second type of autorhythmic cells we have are the conduction fibers, okay, or the cells of the conduction fibers. And these cells are responsible for conducting or transmitting or communicating the action potential throughout the entire myocardium and to the contractile cells to generate the heart or the contractile, the contraction of the ventricles and the atria. All right, so the pacemaker cells generate the action potential. They spontaneously depolarize. The conduction fibers rapidly transmit or conduct that action potential throughout the myocardium. The examples of pacemaker cells are the SA node. SA stands for sinoatrial. That sort of helps us understand where this group of cells are. These are right in the right atrium, right, the wall of the right atrium. And this is the main pacemaker of the heart, the SA node. So the concentration of these pacemaker cells right in the right atrium, and that's the SA node. Second example of pacemaker cells is the atrioventricular node. Right? And this is another congregation of pacemaker cells, but it's going to be in between the wall of the atria and the ventricle on the right side. And we'll illustrate where those cells are in a minute. So that's the SA node and the AV node. These are the two main pacemakers of the heart. The conduction fibers, there are three types here. The internodal pathway. The internodal pathway is the fibers that connect the SA node to the AV node, hence the term internodal. Then there's the bundle of hairs. The bundle of hairs is an intricate network of fibers that runs throughout the septum. And the septum is that area between both ventricles. So that wall that sits in between both ventricles has these fibers called the bundle of hairs. And finally, the Purkinje fibers are going to stretch right up into the ventricle. Now let's look at the rate of these cells before we draw this out. So the autorhythmic cells, we mentioned SA node, AV node, the bundle of hairs, and the Purkinje fibers. Now, as you can see, the firing rate, which is basically how fast are these cells generating action potentials? How fast, how many action potentials per minute? The fastest of those is going to be the SA node. That's like the main timekeeper of the heart, right? The main firing rate of the heartbeat. And that's going to start out at about 70 to 80 action potentials per minute. Okay? The AV node is a little bit slower. That's going to be 40 to 60 action potentials per minute. And then the bundle of hairs and Purkinje fibers are significantly slower in their rate of firing action potential. And we're going to see that this is intentionally done so that there is a delay between the SA node and the AV node. Call that delay the AV nodal delay. It's important that the SA node is firing just a little bit faster than the AV node to allow that delay to happen. That delay ensures that the atria fully contract before the ventricles contract. Okay, that's going to be very important in the heart. You don't want the atria to be contracted or the atria to be contracting at the same time as the ventricles. You want the ventricles to contract after the atria has contracted, and that makes sense. Remember that the blood is moving from the atria down into the ventricles, so the atria must contract fully before the ventricles contract. Also present among these autorhythmic cells are gap junctions. So we'll see gap junctions again. This is on the third or fourth time we're going to see this structure. And it's going to be important here in allowing communication between these pacemaker cells. And this is what keeps the heart beating synchronously, meaning that all the cells are working at the same beat, they're firing at the same rate. So the gap junctions allow communication between the pacemaker cells and keeps the heartbeat synchronous. So the fastest polarizing cells, which are the pacemaker cells, or the SA node in this case, is what really controls the other cells. Right? So the fastest cell, the SA nodal cells, set the tone or set the beat, and the other cells will follow the delay. Okay? Right, so let's take a look at actually, let's keep going here. Now, when we talk about the spread of the action potential between the pacemaker cells, we want to understand, as I was mentioning earlier, that the atria contracts followed by the ventricles contracting. And that's coordinated through the presence of those gap junctions. Okay? Now, the gap junctions are located at a structure called the intercalated disc. The intercalated discs are these small discs that run perpendicular to the fibers of the cardiac muscle, and they're basically the location of junctions between two myocardial cells. If you have one cell sitting next to another cell, that area where they communicate, that area where they connect, that is called the intercalated disc. And the presence of gap junctions, as well as the presence of desmosomes, is very important between these two cells. Desmosomes help to prevent whipping or tearing or any type of stress that's put on these cells as the heart continually empties and fills. So as those ventricles contract and relax, as they empty and fill, desmosomes help to anchor these two cells together very tightly and prevent any injury to the cardiac tissue. Also, gap junctions, as we mentioned earlier, helps to keep the electrical activity happening on the same beat, or help to keep the synchronous heartbeat, keep that material synchronous, or electrical coupling. All right, so let's look at this structure more closely here. So this is an example of muscle cells, and then connected here are your SA nodal cells. So if this is an SA nodal cell, it's going to initiate an action potential, right? Remember that it's autorhythmic. It has the ability to spontaneously depolarize. And then it's going to send that action potential out to the other contractile cells of the myocardium. Now, in between these two cells is this little area, which is called the intercalated disc. And if we look more closely at the intercalated disc, we'll see those two structures that I mentioned. One, the desmosome, helping to anchor these two cells together very tightly. And two, the gap junction, helping to transmit the electrical information from one cell to the other. Okay, two very important junctions between these cardiac cells, and they're located at the intercalated disc. Now, I also want to point out here that if we look at the structure of a cardiac myocyte, we're going to see the same apparatus that we saw with skeletal muscle. We're going to see the sarcomere, right? Remember that cardiac muscle is striated, and because it's striated, that indicates to us that there are sarcomeres. So there's actin and myosin, just like we saw with skeletal muscle, and they are arranged in that parallel formation, just like we saw, which is the sarcomere. Okay, so that's also present in this tissue. And we want to understand that when we start talking about the excitation, contraction, coupling of cardiac muscle. Okay. So let's talk about the conduction system, and let's look at the order of conduction. Okay, so when we speak about the conduction system of the heart, we're really talking about how the electrical activity is transmitted throughout the heart. Okay, now as we know from prior knowledge, the heart has our 4 chambers: right atrium, right ventricle, left atrium, and a left ventricle. Okay. As we just described, there are 2 autorhythmic cells, or 2 pacemaker cells. One is going to be the SA node, and that is going to be at the top of the right atrium. And then we have the AV node, which is in between the right atrium and the right ventricle. Right, the AV node. Now, we also mentioned the bundle of His. So we mentioned conduction fibers. So the bundle of His is located in between the interventricular septum. So this wall that sits between both ventricles. So the bundle of His. It's also called the AV bundle. Okay. And then we have the bundle of His dividing into right bundle branches. So we have right bundle branches here. And then we have this also splitting into left bundle branches. And then finally, the last fiber that we mentioned were the Purkinje fibers. And the Purkinje fibers are in the ventricles themselves, or the wall of the ventricles. So these are our Purkinje fibers. Okay. So let's talk about the electrical activity here. So first thing is that the electrical activity starts in the SA node. So this is where the action potential is generated. And it's going to stimulate the entire left and right atrium. Okay, so it's going to spread throughout the right atrium and then simultaneously spread throughout the left atrium. And this is going to cause both atria to contract. All right, so step 1 here is the impulse is propagated from the SA node to both ventricles, to both atria, sorry. To both atria, and both atria contract. Okay, so that's the first step here. The second step is that the impulse is going to slightly delay before it gets to the AV node. So it's going to get to the AV node, but there's going to be a delay here. We call this step the AV nodal delay. Okay, and as I mentioned earlier, this is important in allowing both atria to contract before the ventricles contract. So the impulse arrives at the AV node, but then there's an AV nodal delay. We do not want that impulse to get to the AV node before both atria contract. That's your AV nodal delay, which is step 2. Step 3, the impulse or the electrical activity is now going to arrive at the bundle of His, which is in between the ventricles. So at this point, it's not fully conducted to the ventricular cells just yet. It's going to the fibers just in between those ventricles. So the bundle of His, or the AV bundle. So the impulse travels to the bundle of His, also called the AV bundle, which is in between ventricles. Okay. All right, so the impulse is going to travel here. All right. Step 4, the impulse is then going to split off to right and left bundle branches. All right, so it splits off into right and left bundle branches. And this is going to be important because it's going to allow the electrical activity to get to the bottom of the heart, to get to the apex, and then gradually move through the ventricles. Okay, so it's going to split off as it gets to the apex, and then it's going to go around to the Purkinje fibers, which is step 5. The impulse propagates through the Purkinje fibers and upwards to the valves. Okay, and here we have the contraction of both ventricles. Okay. All right, now it's important to understand here, both atria will contract simultaneously. Then the impulse will travel in between the interventricular septum, it'll go around to the Purkinje fibers, and then it will squeeze the ventricles up from the bottom. So think about you squeezing a tube of toothpaste from the bottom upwards. That's the same way that the impulse goes from the bottom of the ventricles and moves upwards towards the valve, which helps to squeeze the blood from the ventricle into the vessel that's exiting that valve, that chamber. Okay, and that's how the electrical activity moves through the heart, and this happens every single heartbeat. So every time your heart beats, this entire process happens again and again.